Climate change-induced mass extinctions provide unique opportunities to explore the impacts of global environmental disturbances on organismal evolution. However, their influences on terrestrial ecosystems remains poorly understood. Here we provide a new time tree for the early evolution of reptiles and their closest relatives to reconstruct how the Permian Triassic climatic crisis shaped their long-term evolutionary trajectory. By combining rates of phenotypic evolution, mode of selection, body size, and global temperature data, we reveal an intimate association between reptile evolutionary dynamics and climate change in the deep past. We show that the origin of phenotypic radiation of reptiles was not solely driven by ecological opportunity following the end Permian extinction as previously thought, but also the result of multiple adaptive responses to climatic shifts spanning 57 million years. Hello all, I am UnevenCuttlefish. Today we are going to be looking at the article Successive Climate Crisis in the Deep Past Drove the Early Evolution and Radiation of Reptiles by authors Simões, Kammerer, Caldwell, and Pierce. Released 19 August 2022 in the journal Science Advances. If you have an interest in evolution and radiation of reptiles after mass extinctions, please stick around to learn more. A major challenge in macroevolution and macroecology is understanding the magnitude and duration of organismal response to rapid environmental changes. On ecological timescales, it is well known that organismal diversity, population density, morphology, and life history traits such as clutch size, age of sexual maturity, and longevity can all be affected by shifts in the environment, such as those caused by anthropogenic climate change. However, inferring long-term organismal response to environmental perturbations on geologic or evolutionary timescales is considerably more challenging. Fortunately, the fossil record can provide direct evidence of past environmental crises and how organisms were affected and recovered from such events. The current rate of global warming and species extinction only has parallels deep into the geologic record, thus making our understanding of global climatic shifts in the geologic past especially informative for inferring its long-term consequences for organismal evolution. So what does this mean? It means that every facet of our lives is affected by our environment, within reason, and understanding how the environment influences our existence and to what degree it influences us as it changes is especially important. It is well known that we are in the sixth mass extinction of life on Earth, otherwise known as the Anthropocene extinction. We are losing species faster than we can even catalog them, and knowing what we do about mass extinctions, we can only parallel it to major mass extinctions we have seen in the past, such as the Permian Triassic extinction, or as it is known, the Great Dying. So how do we know what to expect as things continue to rapidly change? We must look into the records we do have. The fossil record. One of the best measures of long-term biological response to a changing environment is the reconstruction of organismal evolutionary rate trajectories, either genetic or phenotypic. These trajectories can be used to understand the adaptive responses of organisms to a changing climate, as evolutionary theory predicts that phenotypic evolutionary rates are tightly connected to biotic, e.g. competition for resources, and abiotic, temperature, environmental variables at both local and global scales through deep geologic time. However, such an approach has rarely been applied to empirical study systems because of the limited availability of data or quantitative analytical tools to explore such questions at broad macroevolutionary scales. Hence, reconstructing the rate of phenotypic evolution across major environmental shifts in deep time is a challenging empirical task, although it provides unique insights into Earth-life system interactions over vast tracts of time. Two of the biggest climatic crises in Earth's history occurred at the end of the Permian. Those events were triggered by unprecedented rates of global warming, culminating in two major mass extinctions: the end Guadalupian, otherwise known as the EGE, at 261 million years ago, and the Permian Triassic mass extinctions, otherwise known as the PTE, at 251.9 million years ago. The PTE represents the largest mass extinction in the history of complex life, with an estimated loss of around 86% of species worldwide. In contrast, the EGE has only been recognized as a major mass extinction in recent decades, but evidence suggests that it was similar in magnitude to other mass extinction in Earth's history, including the Cretaceous-Paleogene extinction. Both the EGE and PTE events reset the entire ecosystems worldwide. e.g., extinction of several groups of early synapsids on land and major reef-building organisms in the ocean, such as brachiopods, and opened up ecological opportunities for surviving animals groups to undergo adaptive radiations during the subsequent geologic periods, the Triassic. Reptiles represent one such group that survived the Permian Triassic environmental crises and adaptively radiated during the Triassic, occupying habitats and ecological roles previously dominated by other groups, such as early synapsids, the evolutionary forerunners of mammals. These factors make reptiles a rare study system to understand the impact and successful recovery of tetrapod faunas in the aftermath of cataclysmic environmental change. However, several aspects of how reptiles respond to these climatic crises remain unanswered, owing to logistical challenges in collecting vast amounts of phenotypic data for fossil vertebrates and in reconstructing organismal evolutionary trajectories in the deep past. Furthermore, such questions are made even more complex by ongoing conflicts regarding fundamental aspects of the reptile tree of life. For instance, there is a strong disagreement between morphological and molecular phylogenies on the relationships of important lineages, which remain hotly debated. These conflicts hamper the reconstruction of a precise timeline of reptile evolution, and yet they remain critical for accurately calibrating major events in their evolutionary history using either morphological or molecular data. Here we address these challenges by constructing a new morphological dataset inclusive of all major lineages of early reptiles, early synapsid, and their closest relatives, early amniotes. This dataset represents the largest taxon sample ever assembled to assess this portion of the tree of life, being 40% larger than the previously largest dataset and the first with the vast majority of all sampled species data based on primary data collection from direct specimen observation and microcomputed tomography scans. We explore this dataset with emerging techniques from Bayesian evolutionary analysis that combine species relationships, diversification dynamics, and evolutionary rates within an integrated and multivariate statistical framework. We further combined our results with data from a body size and global temperature data for over 140 million years, from the late Carboniferous to the late Triassic, to provide a broad overview of the major biological responses toward climatic shifts at this critical interval in Earth's history. This approach enabled us to shed new light on current conflicts in early amniote and reptile relationships and allowed us to establish a precise timeline for the reptile tree of life and the timing of reptile-mammal split, and to answer the following questions. 1. How do rates of phenotypic evolution in reptiles respond to major changes in the temperature patterns during past climate events? And 2. How can the detected long-term patterns inform on fundamental ecological and evolutionary processes, such as selective pressures and organismal adaptability, operating before, during, and after global climatic crises? Our results provide the first instance of agreement between morphology and molecular-based hypotheses of early amniote and reptile evolution, and a substantially more precise estimate for the time of origin of their major evolutionary branches compared to previous studies. We shed light on the primary causes for the historical challenge of placing turtles into the reptile tree of life using either morphological or molecular datasets. Furthermore, we find a heretofore undetected and intimate relationship between long-term global mean temperature trends and the rates of phenotypic, i.e. morphological, evolution in reptiles between the Permian and Triassic. Last, we discuss how reptiles respond adaptively to the end Permian extinction events, which were marked by some of the most drastic environmental crises in Earth's history, finding that ecological opportunity was not the sole driver for the adaptive radiation of reptiles following the PTE. So before we get into what they found, what are they actually doing? Well, they're taking data from every source that they want to put into this model, and it appears to be the largest collection of data ever assembled and used in the framework of any single study. One of the biggest questions this study aims to answer is how do reptiles adapt and become so successful after the EGE and PTE mass extinction events, and provide an answer on how to reconcile disagreements between genetic and visual methods of phylogenetic placement on the tree of life. They are providing us how these groups diversified in relation to global mean trends and the rate of diversification, and they end their introduction statement by saying that the mere opportunity that a certain niche is unfilled is not the entire sole driver of these adaptations, but there's more to the story. This is good because there are people who don't even agree that testudines, or turtles, even belong in the same group as reptiles, as genetically they are more closely related to birds than they are to lizards or snakes. Now, when it comes to why we put them in the same group, well, it really comes down to history. The study of these ectothermic creatures is known as herpetology, and within this study is two groups, reptiles and amphibians. We categorize both groups by several characteristics, but these groups are actually not very related to each other in terms of morphology. We study them together simply because they haven't been studied together in the past. And if you go ask someone who studies snakes about issues amphibians are having, you'll probably get a decent answer, but you won't get as in-depth answer as you would if you ask someone who studies toads, a group that is much more closely related to frogs than snakes are. Respectively, these groups are so highly derived and unique that lumping them together is simply a mistake. Dividing them out is also a mistake. It creates confusion and unnecessary boundaries. But studies like these are important as they help to clear up issues that we've been having for a long time in the study of herpetology. Skipping over a few things that are a little bit more technical than can be explained here in this video, we get to the actual conclusion of this study, stating the long-term organismal response to climate change. Ecological opportunity provided by the demise of dominant terrestrial faunas, the early synapsids, during the PTE at 251.9 million years ago has been previously recognized as the primary driver of reptile adaptive radiation in the Triassic. However, our analysis indicate that a rapidly changing climate during the late Permian to Middle Triassic also greatly affected reptile evolutionary dynamics. In particular, high rates of temperature change coupled with higher global mean temperatures during the early stages of the Triassic triggered a long-term adaptive response by reptiles that was sustained up to at least 15 million years after the PTE. These included accelerated rates of morphological evolution among large-bodied acrosauromorph reptiles, invasion of the marine realm by ichthyosauromorphs and sauropterygians, as well as maintenance of small-body morphotype in lepidiosauromorphs. Specifically among terrestrial acrosauromorphs, their pattern of body size and paleolatitudinal distribution indicates the preferential survival of large-bodied taxa in separate regions, possibly enhanced by migratory events from lower latitudes, and their adaptive radiation in higher latitudes. However, the observed trends in global temperature fluctuations and evolutionary rates do not appear to be triggered by the PTE. It can be traced all the way back to the SAC event in the early Permian at 294 million years ago, or 42 million years before the end of the Permian extinction. This coincides, at least in part, with the time of origin of the major reptile groups, so turtles, marine reptiles, acrosauromorphs, and lepidosauromorphs, in the middle to late Permian. Together, these results suggest a previously unrecognized association between long-term patterns of global mean temperature and its downstream impact on global climate and evolutionary response by terrestrial faunas, including the origin of new clades, construction of new body plans, and geological latitudinal species distributions. Furthermore, our findings shed light on the macroevolutionary process responsible for the origin and diversification of major clades. The emergence of the uniquely diverse reptile fauna that is characteristics of the Triassic, from marine reptiles to chameleon-like trypanosaurs, true lizards, and the forerunners of dinosaurs, is not just the product of ecological opportunity or evolutionary serendipity after the end Permian extinction, as previously thought. Rather, their unique morphotypes also represent the result of sustained, long-term adaptive response to an exceptionally long period of climatic shifts that lasted for 57 million years, one of the longest in Earth's history. So what did they find? They found that the method and evolutionary path that these early reptiles followed was not solely influenced by a simple loss of most life to where they just had the availability to adapt, but rather the ever-changing climate and deep periods of time are just as influential as the immense and dramatic extinction events that happened on Earth. Their results also give direct conclusion on the relationship between groups that were previously not known before. Turtles were an elusive group to understand their evolutionary history and phylogenetic placement, but this study has placed them firmly within the greater phylogeny of currently known lineages. I would like to thank the authors for their hard work and dedication to the field of biology and paleobiology, and I hope you learned something in this video that might pique your curiosity. Until next time.